Hello, everybody. Chapter 18 focuses on gene mutations and DNA repair. There are many topics covered in this chapter, so I broke it down into five different recordings, five segments. In first one, we will start to introduce mutation. What are they? What categories of mutations we recognize? Afterwards, following segments will discuss the reasons, the causes of mutations, followed by segments devoted to transposable elements, and the last segment will discuss DNA repair mechanisms. We will start this chapter talking about Tin Man. Tin Man from Wizard of Oz. Why? What was so characteristic about Tin Man? He did not have heart. And in 1980, when geneticist Rolf Bodmer was studying fruit flies, he discovered a mutation that actually renders this fruit fly unable to live. It was mutation in the heart. Even though the heart in fruit flies is very simple organ, it's just open-ended tube that rhythmically contracts and pumps the fluid. It is necessary for the fly to survive, and this mutation triggers their death. So it's why, because these fruit flies are born without heart, he called it Tin Man. Later on, a similar mutation has been discovered in human and was called NKX2.5. At the beginning, it was not clear what this mutation is linked to. Only later studies revealed that mutations in the NKX2.5 gene, which encode transcription factor involved in heart development, is linked to congenital heart defects. And it was found that about one of every 125 babies is born with certain congenital defect. Some of these defects are very mild and heal by themselves. However, some are deteriorating and they need to be repaired. I'm not sure whether you ever heard about blue babies which are born with a hole in septum which divides right and left heart chamber. Therefore, their oxygenated and deoxygenated blood mix, and that causes a blue color. Now, this disorder is called also Fallot tetralogy. If you are interested in blue babies and the surgery which was developed in Johns Hopkins Hospital by Dr. Blaylock and his technician Vivian Thomas, you can actually watch segment I posted on Blackboard under this chapter, which is from the movie Something the Lord Made. It is a very interesting movie. It's very old, but it's interesting, and you will see what happened during the development of this surgery. They were the first who performed the surgery fixing Fallot tetralogy. So, what are the mutations? Mutations are inherited changes in genetic information, and they play a very important role. When we talk about mutation, we usually link them to disorder, diseases as a bad thing, something bad that happens. However, keep in mind that genetic mutations are source of genetic variation, and basically genetic variation is necessary for natural selection and evolution. So mutation in long run play a role in the evolution. Nowadays, we also explore or exploit induced mutation to study function of particular genes. So they are a very important probe in fundamental biological processes. Let's first start to discuss categories of mutations. And in multicellular organisms, we can distinguish between two broad categories: somatic mutations and germline mutations. Somatic mutation arise in somatic tissue. So, what does it mean they occur in non-reproductive cells, and they can be passed to new cells through mitosis, which creates a clone of cells which are having mutation, like you can see outlined right here. It is estimated that our body is formed by approximately 10 to 14 cells, and in every million cell division one mutation usually may arise. Many of these are not detected, many are corrected by DNA repair mechanisms. However, if these mutations are not corrected and they affect cell cycle, if they affect genes that control cell cycle, they can trigger cancer. However, somatic mutations are not passed to further generations, so they cannot affect the evolution. Germline mutation, in contrary, arise in cells that produce gametes, and therefore during meiosis and sexual reproduction, this germline mutation can be passed to approximately half of the members of the next generation who will carry this mutation. It is outlined right here. So they are a past germline mutation to offsprings. They are those which actually can change allelic frequencies within the population, which can introduce higher variability, genetic variability, and can serve as a base for selection and the evolution. There are several ways how we can classify gene mutation. The classification can be based on the molecular nature of mutations, on the nature of the phenotypic effect of the mutation, based on the causative agent of mutation, and so on. So we will start to discuss types of mutation based on their molecular nature. By the molecular nature, we can divide mutation to base substitutions, insertion and deletion, and expanding nucleotide repeats. We will go into more detail to this classes of mutation in following slides. First, we will discuss base substitution, transition, and transversion. So the simplest type of gene mutation is a base substitution. It's outlined right here. If you substitute one nucleotide for another, you will affect one codon. So one codon will be changed. What does it mean? In polypeptide, one amino acid might be changed or replaced by another. Base substitution, which alters single codon, may happen via transition or transversion. In transition, purine is replaced by purine, or pyrimidine is replaced by pyrimidine. In transversion, purine is replaced by pyrimidine, or pyrimidine is replaced by purine. There is more transition usually than transversion. Transition arises more frequently because transforming a purine into different purine or pyrimidine into different pyrimidines is easier than transforming purine to pyrimidine and vice versa. Now keep in mind base substitution, whether it is transition or transversion, affects one codon. One example, sickle cell anemia. Sickle cell anemia is transversion, and we will talk a little bit more about it in next slides. Another group of the gene mutation based on their molecular nature are insertion and deletions. Insertion or deletion of one or two nucleotide may have deteriorating effect because it causes frame shift mutation. If you insert or delete one nucleotide, you shift reading frame, you change reading frame. So every nucleotide, every amino acid after this mutation will be different because the genetic code, following genetic code, would restore reading by 3 and it would be shifted. So let's have some example. Let's say if you write as a sentence which contain words which are read in 3 letters, such as cat, has, hat. Now, imagine that you delete one letter, one nucleotide. Let's say you delete T and you restore reading in 3. You know, genetic code is in triplets. So what will be it? What it will be now? Kh, ASH, AT, which doesn't make any sense. You can see here. And this is what happens when you insert or delete one or two nucleotide. You change reading frame, you change coding. So basically codons after this mutation will be changed, so they will encode the different amino acid and the polypeptide will be changed. Now if there is so-called in-frame insertion on deletion, the reading frame can be restored. What does it mean in frame? You delete or insert 3 nucleotides. It's like here, what if instead of deletion T, you delete has? So you can still restore it partially, cat, hat. So the sentence is not fully understandable, but you can at least see that it refers to cat and a hat. So it is this kind of mutations are called in-frame insertion and deletion because they delete or insert 3 nucleotides, so the reading frame after this insertion or deletion is restored. So here you have outline in more details of transition and transversion, where in transition adenine can be exchanged for guanine or guanine for adenine, and pyrimidine by pyrimidine, thymine by cytosine or cytosine by thymine. Transversion can actually replace purine by pyrimidine or pyrimidine by purine, so there are more options outlined right here. As I mentioned before, sickle cell anemia is a transversion. You know that in sickle cell anemia only one nucleotide is substituted. It is thymine which is substituted by adenine, so it is transversion, which converts the genetic codes, and reading of the genetic code replaces glutamic acid for valine, charged for non-charged amino acids. So this one transversion completely destroyed functionality of hemoglobin. Transition, you remember we talked about Tinman mutation NKX2.5. So 4 different transitions have been detected within this gene, which might alter or cause congenital defects. The last category of mutations based on the molecular nature we will discuss are expanding nucleotide repeats. Expanding nucleotide repeats trigger increase in the number of copies of set of nucleotides. One of the best studied or characterized disorder which result from expanding nucleotide repeats is fragile X chromosomes. This is associated with this characteristic constriction on the long arm of X chromosomes, which represent a fragile site. So how these nucleotide repeats can expand? It is outlined right here on the right side. Let's say the DNA molecule has repeat CAG. So there are several CAG repeats in this DNA. When DNA replicates and you have new strand which is replicated, because of the repeats it can form hairpins. If the newly strand form hairpins, the part of DNA, the template, where the hairpin forms on the newly synthesized DNA will be replicated twice and will increase the number of repeats on the newly synthesized strand. So when this newly synthesized strand, which has higher number of these repeats because of the hairpin formation, will replicate second strand, you will come up with new DNA molecule which contain additional copies of the CAG repeats. The number of copies of repeats correlate also with the instability of this nucleotide repeats. So more repeats, more instability is introduced. And this instability is increased with each new generations, which we discussed it is an anticipation. There are several disorders which are actually caused by expanding nucleotide repeats, and they are summarized in the next slide. This table just shows you some examples of human genetic diseases caused by expanding nucleotide repeats. You don't have to memorize them, just that you know that this genetic disorder has very high impact. And for example, in Huntington disease, these expanded repeats produce a toxic protein which has extra glutamine residues, and toxic protein in brain can cause a lot of damage. So just look at these examples and reflect how important these mutations can be, what kind of deteriorating effects they can have.